Hiroshi Hamada is the director of the Riken Center for Developmental Biology in Japan, which is a leading center for the field, where he's also the team leader for the organismal patterning team. He holds an MD and a PhD, and he's held positions at NIH and Newfoundland in Canada, as well as Tokyo, Osaka. He's had a long career, which spans subjects of many scales, and it's included landmark discoveries, including the discovery of OCT4 and of Lefty, which we just heard about. He spent recent years studying the left-right asymmetries in the development of mouse embryo, in particular looking at the roles of both the motile cilia that we saw in Daniel's talk, as well as the immotile cilia, which are involved in the sensing of this flow. And so I think we're going to hear more about that second class right now. Take it away, Hiroshi.
Okay, thank you. Thank you very much for the very kind introduction. Maybe this is the kindest introduction I ever heard. But anyway, I'm not a director anymore, so I stepped down from director. Yeah, that's okay. So I'm
former director.
Yeah, I'm enjoying the life in the lab these days. So thank you very much for the inviting to this very unique meeting. And I also like to talk about the left-right asymmetry. And thanks to the previous speaker, Dan, maybe I can skip the introduction before, maybe I can skip most of the slides. But maybe I will show you that to remind you the same conclusion from a slightly different angle. So that invertebrate left-right asymmetry is established by three consecutive steps. The first is asymmetry breaking, and the second, which is followed by differential patterning of the cell on the both sides by the left side determinant nodal and the lefty, and finally the asymmetric organogenesis of the visceral organs. So, but today I'll focus on the first step, which is symmetry breaking. So, first, today I would like to talk about cilia-dependent mechanism in the mouse. In particular, I'd like to focus on how mouse embryo sense the fluid flow. And if I have a time, maybe I like to introduce the cilia-independent mechanism in reptiles. So the first, the symmetry breaking in the mouse embryo. So you are on the left side, you are looking at the mouse embryo about 8 days after fertilization. This is a left lateral view. And as Dan said, the symmetry breaking takes place at a region called node, which is here. And this is a very small cavity located at the midline. And if you look at closer, there are about 200 cells at the bottom of the cavity, and from each cell, each cell has a single cilia. And somehow all the cilia rotate into the clockwise direction, and this rotational movement can generate the unidirectional fluid flow. And this is actually the amazing discovery made by Nonaka-san and Hirokawa sensei many years ago, maybe 20 years ago or longer. So the rotational movement of the cilia can generate the unidirectional flow instead of the vortex, because the cilia is tilted toward posterior side. So I like to remind you that the left-right, well, we have three kinds of the body axis, but left-right axis is the last axis that is determined during the development. So embryo wisely use the pre-existing information to perform the left-right symmetry breaking. So as also this also Daniel, as Daniel said, the first information is anterior posterior axis, which is represented by the posterior tilt of the cilia, and also second is the dorsoventral axis, which is represented by the direction of the extension of the cilia. This is a ventral and this is a dorsal. And finally, the chirality of the cilium, which is here, the direction, the rotational chirality of the dining arms, which probably determine the direction of the rotation. So this is really the beautiful, I think it's the beautiful system to break the symmetry by using the pre-existing information. So I think it's we have learned a lot about how to generate fluid flow by the motile cilia, but we don't know much about the remaining question is that we don't know much about how the embryo sense the fluid flow, and this is the big long-standing question since the discovery of the flow, and almost 20 years, more than 20 years ago. Still, there are two possibilities has been proposed. One is a chemosensing, and the other one is mechanosensing. So that's we still don't know which is correct, and so so I like to, but we don't know which is correct. However, we do know a little bit about it. At least we know that it is an immotile cilia that is sensing the flow. Mechanosensing or the chemosensing, whichever, the immotile cilia are sensing the flow. So that so you are looking at here, you are looking at the node, and this is the periphery of the node, and the immotile cilia are genetically labeled. As you see here, these are immotile cilia. They are not rotating, they are just fluctuating. And so these cilia, immotile cilia, are sensing the flow. So this is a summary of the current our understanding. So that this is the node, the flow comes, the flow is generated by the motile cilia located at the center of the node, and at the periphery of the node, there are immotile cilia, they are sensing the flow. And by sensing the flow, probably, as Daniel said, the calcium get into the cell by using the PKD1 like 1 and PKD2. Then that final target is a degradation of the one particular messenger RNA called DAND5, or as the same gene but a different name. That I'll be using the different name, but servers like to messenger RNA. This is the target of the flow. So the calcium has been implicated in the symmetry breaking, and we know that the calcium is inhibitor. For example, inhibitor experiments suggest that calcium is essential to set up the asymmetry, but nobody has ever looked at the calcium directly, except that the Xiaolu in MGH at this moment, he has detected the calcium in zebrafish, the organizer, but at least in a mouse node, nobody has looked at, well, it's not clearly look at the calcium in immotile cilia. So this is also that this is a little bit a controversial issue, but finally we have been able to, the Mizuno-san, Katsutoshi Mizuno-san was able to detect the intraciliary calcium influx at the node. So we have established the transgenic mice that express the different calcium sensors. Here we have two examples. This one can monitor the ciliary calcium, and the bottom one, this one, can monitor the ciliary calcium and also at the simultaneously the cytoplasmic calcium. By using this calcium sensor, and we expressed this calcium sensor in the immotile cilia of the perinodal cell, specific by using the cell type specific enhancer element. So then this is you are looking at the calcium at the periphery of the immotile cilia. So as you see, there is an active calcium transient, especially in the left side of the node under this. However, after careful analysis, we realized that these transients are actually the mixture of the two types of the transient. One is cilia independent calcium that probably comes through the cytoplasmic membrane. Then it will come, or the dilute. It will also come to the after amplified in ER, the calcium comes to the cilia. So this is a one type. But this type of the calcium transient are not asymmetric. It's bilaterally equal. However, the second class type of the calcium transient are cilia dependent. So it's probably the intraciliary the calcium influx. And this one is flow dependent, the PKD2 dependent, and also it's asymmetric. It's preferentially local, found on the left side. So one can distinguish the two types of the calcium transient by using the specific inhibitor. For example, tapsgargin. This type of calcium transient are sensitive to tapsgargin, but the real one, the cilia dependent, the asymmetric calcium transient are resistant to the tapsgargin. So one can distinguish. So let's look at the cilia dependent tapsgargin resistant the calcium transient in the immotile cilia. So now this is you are looking at the tapsgargin resistant ciliary calcium spikes here and also here. As you see, the most of the oscillating cilia, immotile cilia, are localized at the left side, and in particular, the left posterior side of the node. So this is a summary. So Mizuno-san looked at many embryos, and this is a summary of the location of the oscillating immotile cilia. As you see, that these cilia are localized at the left posterior side of the node. And this is very important because the left posterior region is exactly where the first molecular asymmetry breaking takes place at the node. So that here on the right side, you are looking at the phosphorylated SMART2, which is the downstream target of the flow. And so this phosphorylation begins in the very small area located at the left posterior side, then expand on the left side. So this side is the left posterior side, exactly the same side, location of the oscillating cilia. So we think it's the, yeah. So this is an important point. So not only that, this intraciliary influx is essential for the left-right asymmetry at the node. So, for example, one can inhibit the calcium transient by expressing the purple albumin, which can trap the inhibit the calcium. Then you can we express this purple albumin in the same immotile cilia. Then the molecular asymmetry at the node is now impaired. So the suggesting that this calcium influx is essential for left-right asymmetry. So what about what happened to the cell when they received the flow signal? And this is also the question that Daniel addressed. And so the target of the flow signal is the messenger RNA encoding the DANDI5 or Sabras-like 2. So this messenger RNA is bilaterally equal before the flow comes, but when the leftward flow comes, this flow will degrade this particular messenger RNA on the left side. So then at the end, as a result, the level of messenger RNA is higher on the right side. So this is a readout of the flow. So, and this regulation is not at the level of transcription, of course, but its regulation takes place at the level of post-transcription. So it's involving the 3' UTR of the Sabras-like 2. So, for example, if you replace the 3' UTR endogenous the 3' UTR of this gene by a different heterologous the poly 3' UTR for such as SV40. Now you lose the left-right asymmetry. So it's clear that the 3' UTR is important. So then we wanted to know the molecular mechanism of the flow-mediated messenger RNA degradation. So to monitor the degradation easily, so we set up another transgene, which is composed of, which encode the destabilized Venus plus Sabras-like 2 3' UTR that can respond to the flow. And this cassette is driven by a clown cell, the specific enhancer element. So it can express, it will express in the immotile cell with the immotile cilia. So as you see, this is a pattern of the fluorescence given by this transgene. At earlier stage, the level is bilaterally equal, but when the stage goes on, now that the right side is much stronger than the left side. And this is exactly similar pattern of the endogenous Sabras-like 2 messenger RNA. So suggesting that this transgene can recapitulate the pattern of the level of the Sabras-like 2 messenger RNA. So by using this transgene, we examine if the transgene can respond to the flow or some PKD2 mutation. So this is the wild type situation. The right side is much stronger, but in case of the IV mutant, which lack the flow, now the asymmetry is lost, and also PKD2 mutant, the asymmetry is lost. So the 3' UTR action requires the flow and PKD2 calcium channel activity. So after this is a summary of the molecular, the the molecular mechanism, so that we have been able to map the flow responsive element in the 3' UTR of this particular messenger RNA. We have about at least about 200 base pair sequence that is conserved among the vertebrae. The sequence is recognized by RNA binding a protein called BIC1, and BIC1 will make an interact with another protein complex called C03, which is the messenger RNA degrading the enzyme. So when flow comes, the calcium that will activate, you will have a calcium in the cell, and somehow the calcium will activate the BIC1 and C0 and bring this complex to this flow responsive element of Sabras-like 2 messenger RNA, then induce the degradation of this particular messenger RNA. So that's we think it's happening in the immotile cilia. However, going back to the original question, the original question was, how does the embryo sense the flow? Is it chemosensing or mechanosensing? Our data, current data, they don't tell you anything about it. They cannot distinguish. So I think it's by using the materials and transient, we have been able to address this question. Well, I think it's at this moment well, we don't know which model is correct, but for several reasons, I thought it's the mechanosensing is more likely. So we wanted to test this mechanosensing model by using the optical tweezer. So this is a collaboration with the biophysics people, and Takanobu Kato-san. Kato-san is moved to my lab, and he's now doing the optical tweezer in my lab. But the idea is very simple: just impose the mechanical stimuli by using the optical tweezer and see if the cell can respond, and then see the calcium response, and also the response of the Sabras-like 2 decay. So Kato-san has been doing this kind of experiment, and still it's ongoing. But so he imposed the force, mechanical stimuli into this direction, this or this, so that probably the physiological direction. Then after that, he looked at the calcium, the response, and also the level of the Sabras-like 2. And what happened is, yes, that by imposing the mechanical stimuli, yes, there is a calcium response, and also the Sabras-like 2 messenger RNA undergoes the decay. And I just want to show you some typical response of the calcium, intraciliary calcium, and this is time 0. You impose the mechanical stimuli, and typically after the 10 or between 10 and 30 seconds, then we will get the nice calcium response in the cilia. So we still don't know. We have to. I think it's one problem is why do I have a long drug? of that 30 second. It's why is not the immediate response? That's, we don't know. We still have to solve the several questions. But in any case, the cell cilia are responding to the mechanical stimuli. But probably the more important thing is the surplus like to messenger RNA decay that takes place in response to the mechanical stimuli. So that's really the encouraging to me. So do I have a time?
Yes, we have a few more minutes for the talk portion, and then there's a whole another 15 minutes for discussion.
Okay.
And I'm sure many people would be very interested in this reptile and bird discussion.
Most of the data are published, but I only have one unpublished slide. So, but I think it's that we were interested in the reptile because that the mechanism is the diverse among the vertebrate, especially the symmetry breaking. The mechanism is different, and I guess that many years ago there were two reports suggesting that the chick embryo can establish the reflex asymmetry without motile cilia. So we wanted to know if this is true for the reptile, and also we wanted to know the mechanism, why they can establish reflex asymmetry without motile cilia. So there must be some new mechanism. So maybe I should skip the point is that we look at the embryonic region equivalent to the mouse node, and there is no motile cilia. And however, the key most important observation was nodal expression pattern at the node. No, I shouldn't say it's node, but left-right organizer, as you use the left-right organizer. In case of mouse, nodal expression at left-right organizer is bilaterally equal. And however, the dandified or surplus like-to expression pattern become asymmetric. That's how you can, the mouse can create the left-right asymmetry. However, in case of reptile, nodal expression itself is already asymmetric. The left side is much stronger than the right side. So this will and also what happened to the surplus like to dandify? Actually, the dandify gene is lost during evolution. So reptile doesn't have it. So that's understand. It's easy to understand. Well, these reptile, they don't need a flow. They don't need motile cilia. They don't need a surplus like to target, because the nodal expression is already asymmetric from the beginning. So that's the chicken reptile and mouse genopath fish, they use a quite different different strategy. This is a common strategy. Common part. It's the nodal expression in the lateral plate mesosome, it's the left side, and also the left, the organizer region, nodal activity is asymmetric. However, the chick, the nodal expression is already asymmetric. In case of mouse, nodal expression is symmetric, but surface like-to is asymmetric. As a result, nodal activity become asymmetric. So, well, this is a common part, and this is the different part. So my big question at this time is, why is nodal expression asymmetric at left-right organizer? So what's the mechanism? So one way to look at, to understand is we are trying to examine the transcriptional regulation of chick and reptile nodal gene by searching for the LRO specific, the enhancer. So this is a reptile nodal, and this is the chick nodal, and this, we have the exon and also couple of enhancer, conserved enhancer element. We chop down the genomic region to the about 5 kilobases, then make the reporter plasmid, and then electroporation to the chick embryo, and then trying to search for the enhancer responsible for asymmetric expression at the left-like organizer. So this is one of the construct, at least showing the asymmetric expression in the lateral plate mesoderm. And also probably it looks like a left-like organizer, but it's not, at this moment, this is a new, very new data, and we have to be careful about if there is a asymmetry, left-like asymmetry in the expression. So in any case, we'll look for the LRO specific enhancer element by this method, and then maybe I still have a 2.5 years to go before retirement, and hopefully I can have some clue for this. So anyway, thank you very much for your attention. That this work is. I'd like to thank my former colleague in Osaka University, and also the current member in my lab in Riken, Kobe. So thank you very much for your attention. Thank you.
Thank you. We have some time for questions. So we have one from Boris.
Thank you very much for a wonderful talk. So the quick question is, these mechanosensory cilia, are the ones in the node very different from, say, the ones in the kidney?
That is a tough question. In general, we think it's similar, but actually we never look at the structure of the immotile cilia, and I think we've been trying to examine the fine structure like by the electron microscopy, but the number of the cilia are not many. So actually, and the quick question is, we don't know. Probably similar, but
I just remember reading that even MDCK cells have these cilia, and the claim is that even a single cilia can generate detectable calcium current. Well, thank you.
Yeah, thank you.
A related question. So is the structure between the motile and immotile cilia known to any degree?
Yes, I should have, I hope I should have the slide, but the biggest difference between the motile and immotile cilia is the presence of or absence of the motor protein complex called outer dining arms or inner dining arms, and that can be seen by electron microscope. So that's the biggest difference.
Yeah, I guess I'm curious, is there more difference other than the dining complex, or is it just, whether it's being driven or not?
That could be I don't know, I think it's the immotile cilia at the node, my collaborator was estimated the sensitivity of the mechanosensitivity of the immotile cilia at the node, and this is based on the theoretical and also the observation. It seems to be very sensitive. I forgot, but probably the 2 piconewton is sufficient to sense the flow. Mm-hmm. And so that I don't know what this means, 2 piconewton, but he said that's this cilia must be very sensitive. So it must have some unique structure, but we don't know that. Yeah.
We have a question from Sigali.
Hello, Hiroshi.
Hello.
Very nice talk. I enjoyed it very much. I have a sort of general question because, of course, in the wild type animals, what happens is in the node is an on-off response. So you get nodal on the left side. But of course, in experimental situation, in some mutants, the pattern of expression of nodal, at least in the lateral plate mesoderm, can be a bit more complex than being on the left or the bilateral. Sometimes the bilateralization is not exactly at the same anteroposterior level. So I was wondering, do you think some of this could be due to some sort of incomplete activation of the system in the node, or is this only due to something happening at the level of the lateral plate mesoderm?
Right, I think you're right that if you look at the nodal expression pattern, for example, and organogenesis, there is a discordance. You cannot predict the organ laterality by looking at the nodal expression in the lateral plate, and also nodal expression at the node. It's the gene expression pattern does not really predict the organ phenotype, and that's, I don't know. I think you may be right, or maybe that there may be some I don't know, compensation. There is a compensation mechanism. Even the asymmetry is very small. The embryo has a mechanism to amplify the difference. So when you do that, the in situ hybridization, it looks like there is no expression, because the level is so low. However, maybe there is a difference, already a small difference on both sides. So I'm sorry, I don't have a good answer to you. So that's something that you have to solve. Please.
I have a small question. Do you know, Hiroshi, if the mechanosensitivity of the cilia, is it isotropic, or if you have, say, flow from left to right, they would respond strongly than right to left flow with respect to some AP axis?
Could you, right. You mean the optical tweezer experiment?
Yeah. If you pull it upward, would it respond the same way as?
Yeah. Actually, it was technically a little bit different to impose a different direction. So the physiological direction was the easiest direction to perform. So actually, we never did it like this way or that way. So sorry, we don't know that. So, maybe we should try, but yeah, that's a good point. But so we should try.
So just to be clear about the physiological direction, meaning so it's tilted towards the posterior and moving laterally. That's the easiest direction. Is that right, Hiroshi?
Yeah, that's
and
correct.
So it would be stiffer to change the angle along the AP axis.
Yes. Yeah. So the flow is coming. The flow comes this way. So this is the physiological. I think it's the direction. But in this particular experiment, we are using the mutant embryo, lacking the endogenous flow. So, but anyway, so this is this is a physiological direction.
Can I interject a question?
Yes, please.
In your studies of the birds and reptiles, you were looking at transcription factor regulation for the nodal gene. But I'm wondering, I believe that the right-left asymmetry has to somewhere derive from a chiral molecule. So have you looked at these cells to see if there's any chiral structure in them anywhere?
Right. That's the good point. And right. We don't know that. But in some other the organisms, such as snail or the Drosophila, I think it's the actin and the myosin, and they place they seem to show the chirality at the level of the single cell, within the single cell. And these actin and myosin are the plates are essential for setting up the symmetry. But in case of reptile, yes, the vertebrate, we don't know that. Yeah.
Okay, thank you.
But maybe the myosin, yes, myosin. In case of zebrafish, the myosin is essential. There is a report that the myosin mutant that shows the disrupted the asymmetry. But in case of the mammal, there are many myosin, but one particular myosin class, which is equivalent to the zebrafish, the myosin. But there's a mutant, the mutant rat, not a mouse, but they show that the normal laterality. So maybe that myosin is not playing alone, but we don't know that.
Excellent. So if there's one last quick question, we could field that before going on break.
Can I ask a question, Noah?
Yes, please, to know.
So, Hiroshi, you showed that there's this element within the 3' UTR that's flow responsive, and you identified a couple of proteins that bind to that site. I was wondering if those proteins contain like calcium binding motifs, or if there's any, clear regulatory domains within those proteins that would connect them to the calcium influx that you're seeing. Yeah, that's the point. Yeah, that's something I really like to know. But unfortunately, these proteins, the big one and the C0-3, they don't have that kind of domain. But I guess that the calcium is somehow modifying, activating the enzymatic activity or the interaction between the protein by, for example, phosphorylation. I don't know. So we are trying to, well, I've been collaborating with Daniel Constant, who is the expert of the big one, and so as a collaboration, we're trying to find out the role of the calcium, what it's doing, and is it phosphorylating the protein or this? But we don't know the answer yet. Yeah, but that's something I really like to know.
Thank you so much to both of our speakers. So